In this video, we're going to look at the mitochondria. So far, we have gone through glycolysis and we have gone through the citric acid cycle. And through these processes, we have seen the creation of ATP and GTP and these NADHs and FADH2 molecules. Now, these molecules are how we get the majority of energy from breaking down glucose. We're going to take the electrons that we've gained in these compounds and we'll pass them into the mitochondria's electron transport chain. From there, we're going to transfer the electrons through a series of redox centers to ultimately get to oxygen to make it into water. As we are transferring electrons, we're going to expel protons from the mitochondria, creating a proton gradient across the inner mitochondrial membrane, which we will then use to create ATP. So before we look at oxidative phosphorylation, let's look at where it's happening at, the mitochondria. This contains your pyruvate dehydrogenase, citric acid cycle enzymes, fatty acid oxidation, electron transport, and oxidative phosphorylation enzymes. You have a smooth outer membrane and an invaginated inner membrane, with the invaginations called cristae. The number of cristae depend on what type of cell you're in. And this inner membrane divides our mitochondria into two different compartments, the inner membrane space and the matrix. Matrix is where you have a lot of oxidative metabolism enzymes, substrates, cofactors, ions. This is where your DNA, RNA, and ribosomes are located for the mitochondria. But of the 1,500 proteins in the mitochondria, only 13 are made in the mitochondria itself. That's because these proteins are super hydrophobic, and so they couldn't be transferred from the cytoplasm to the mitochondria easily. Now, the cristae themselves, they can be simple tubes or very complicated assemblies. And these cristae allow the formation of microcompartments that restrict the diffusion of substrates and ions between intercrystal and intermembrane spaces. This means that we have differences of pH that we can use in oxidative phosphorylation. So let's look at the membranes a little more in detail. So the outer membrane, we have a lot of pores there, and this allows the diffusion of molecules up to 10 kilodaltons, which means our intermembrane space has more or less the same type of ions and metabolites as our cytosol. The inner membrane has a lot of, and that's shown right here, the inner membrane has a lot of more proteins than the outer membrane. And we can see that through the number of particles per micrometer here. So we have a lot of particles on the interface of our inner membrane. And only oxygen, carbon dioxide, and water are freely movable through this membrane. For ATP, ADP, pyruvate, calcium, and phosphate, we have specialized transporters. And this high regulation of movement allows an ion gradient to be created, and it also allows compartmentalization of different functions between the cytosol and the mitochondria. Now, for oxidative phosphorylation to happen, we need NADH to get in. And so we have different shuttles to allow NADH from the cytosol to go to the mitochondria. One way we already looked at is the malate aspartate shuttle, and we talked about that a few weeks ago. Another way is the glycerophosphate shuttle. Here, 3-phosphoglycerol dehydrogenase catalyzes the oxidation of NADH by DHAP to yield NAD+. Remember, we've seen dehydrogenases already. When you see the word dehydrogenase, you should suspect NADH or FADH2 to be involved. Now, when we take this NADH and make it NAD+, that can go back into glycolysis, and the electrons that we have taken from this NADH, we give to FADH2, and that will enter the electron transport chain as well. Now, to get ATP and ADP in and out of the mitochondria, we have a specialized transporter for that. Otherwise, it would not make sense to make all this ADP in the mitochondria and have the cell not be able to use it. So the inner mitochondrial membrane, that's where our transporter is located. Transports ATP out of the matrix and transports ADP in. This is a dimer, 300 residue subunits. Each subunit can bind ADP and ATP. So two different conformations. An ATP-ADP binding site for facing the matrix and one facing the intermembrane space. And this transporter will change conformations when you're binding a ligand. So what happens is that every time you import an ADP, you export an ATP. So you're doing a one-to-one trade. The phosphate, for sticking a phosphate onto ADP, we have a phosphate carrier for that, a hydrogen phosphate symport, and that's driven by our change in pH, or our difference in pH that we create through our proton gradient. And that is it for this video. I will see you in the next one.